Okay, so our last speaker this morning is Dr. Matthew Hall, who is a branch chief of NIH National Center for Advancing Translational Sciences. And he's going to talk to us about the prehistory and future of platinum anti-cancer drugs. Welcome.
Thank you very much. Thanks, everyone. It's a tremendous pleasure to be here and to meet some of my Cisplatin heroes for the first time as well. I've read all the papers, and now I get to meet the people. So Jim had asked me to speak a little bit about the prehistory of platinum-based complexes and to also address some of the future based on some writing I'd done recently. I was amazed to see that license plate earlier on. I got the Twitter handle. He got the number plate. So it's a real pleasure to be here. The other thing I didn't realize when I was asked to speak as an NIH person who's done a lot of work at NCI with experimental therapeutics, what the mood of the room would be about NCI. So I might regret one of the slides that I've got here. We'll see how we go. So we're going to go quite a way back. This is Michele Peyrone. My Italian friends in the audience tell me that I've totally butchered the pronunciation. But he was born in 1813 and he lived to 1883. And I guess if he'd wanted to, he could have filed the composition of matter patent for cisplatin. So he was the first person to synthesize cisplatin. He didn't quite know it. It really took Alfred Werner to demonstrate the isomeric nature of cisplatin compared with transplatin and Magnus salt. He grew up in near Torino in northwest Italy, and lived most of his life there, though he traveled a great deal. We believe he was quite wealthy, although he did have quite a tragic life in spite of his wealth. And as you can see here, these are just a few miles apart. He was born in Mondovi, spent his final years in this tiny little village of Maglione Alpi, and died ultimately in Caru, where his daughter lived. So he did his work on platinum in Germany, and this is just sort of a snapshot. Mike, I wanted to get some old people pictures in here as well. So this is Magnus, who generated Magnus green salt, which is also isomeric in terms of molecular formula with cisplatin as well, if you break it down. And Magnus had produced this complex that others had picked up and started studying. Before Peyrone got to von Liebig's lab, where Peyrone did all of his work in Gießen in von Liebig's lab, Reisert had published a molecule called Reisert salt, which we now know to be transplatin. And so when Peyrone arrived, he started working on platinum complexes. Von Liebig was a famous agricultural chemist, so what was he doing playing around with platinum complexes? I've been communicating with quite a few people. I think the answer lies in the Kaliapparat, which is probably another word I'm going to butcher today, which is actually the 5-chambered vessel you see on the American Chemical Society's logo. It was used as part of the first elemental analysis or elemental composition experiments that were performed. And von Liebig's group were using platinums to react with alkaloids, with amine-containing molecules. And so I think in that context, he'd asked Peyrone to come to his lab and to investigate the reactivity of platinum. And so all Peyrone was trying to do at the time was to take platinum 2 chloride, add an excess of ammonia and make Magnus's green salt. But maybe a little bit like Rosenberg and his companions, he noticed on occasion that he could see flecks of yellow powder in amongst his green formulation. And when he took the time to separate these out, he found another isomer of this platinum complex, which was called Peyrone's chloride or Peyrone's salt, depending on where you read. And he wrote it up, and it was the good old days, so he actually published the same paper in 3 different languages, and that counted as 3 publications. So it was a great time to be alive. Another tip for some of you out there chasing tenure. Ultimately, Peyrone returned back to Italy. He was a physician by training, but became an agricultural chemist, and I think really became the evangelist for von Liebig in Italy. And he wrote a beautiful book on agricultural chemistry that he published around 1871, ultimately deceasing. And this was I think if you could sort of get a sense of the obsession, I think this slide with his tomb is probably it. Unfortunately, I haven't been there yet. In Mondovi, there's a monument to Peyrone on the wall. They actually ripped out a fresco to put this up when he died. So it gives a sense of the respect that his local community had for him when he died in 1883. And this is where he's buried, and I interacted a little bit with the council at Caru to try and track this down. And the probation officer who went in for me to try and take photos said the crypt was very dark and it was hard to read the names, but the grave was unadorned. So if this is unadorned, I look forward to having an adorned grave when I die. This is actually the tomb of his daughter's family. He married the mayor of the local town. And I love this quote. This is from his first paper on cisplatin. Peyrone wrote, "I'm determined to pursue the subject with all my energies without having regard to difficulties at every step in an expensive, which it is, and delicate research." And I think that really resonates with some of the presentations I heard this morning about cisplatin. So we've all seen this slide, and I think you've all seen these images in slides as well. I did a lot of work when I was in Sydney on platinum fall complexes, and then moved to the National Cancer Institute to study resistance towards these agents. And of course, these slides I inherited from Trevor Hampley, my PhD mentor, and as beautifully as they capture the narrative and the chronological narrative, they don't necessarily capture a lot of what we've heard about today, which is some of the challenges associating with advancing a compound like this to the clinic and working in a really complex team science environment, like the way it happened. So I promised Jim that I'd sort of dive into the NCI archives, and which I did. I have no idea if I'm even allowed to show this stuff. I didn't know we'd be being web streamed, but anyway, this is the earliest reference I could find to cisplatin. It turns out that minutes from meetings in the federal government are really boring. So I captured a few things here. This is October 1, 1968, and this is the first entry for cisplatin, which was called 11975. This is the structure as it was drawn, which I was horrified to see, but I've actually seen it today on a few slides as, I guess, an early representation of the way cisplatin was drawn. There's a couple of things that was received from MSU to interview with cell wall synthesis, and this is a committee deciding whether to take on cisplatin and advance it into animal testing. And they make a couple of comments. It's $47,000 a kilogram. It's really easy to synthesize. It only takes a week to make 10 grams. I'll be interested to find out at lunch if that's true. Solubility was noted as well, and they have activity against bacteria. And it passed decision point 2, and the reason it passed decision point 2 is because it's a compound which is a new chemical class that had been demonstrated to have anti-tumor activity, which is exactly what the National Cancer Institute was looking for at the time. So I don't know if these meeting minutes match up with the personal experience of trying to push these compounds with the NCI into testing. They certainly did go into testing, and Zubrod, who did a lot of early work with cisplatin at NCI, said in this meeting he made a commitment to test any compounds that Dr. Rosenberg would provide. So it's interesting to see the internal discussions, which of course are very dry and recorded here, and probably intended to be private in terms of minuting. So that's sort of the context, and it's really interesting to look at that. So that's 1968. That's before the compound had gone into humans, which occurred in 1971. This is a great figure from a Lloyd Kellen review, who really should be at this meeting. It's quite sad. And then, of course, it was approved in 1978. But in between this time, you would think, okay, NCI takes it, 1978, it's approved. And the first patient saw this drug in 1971. So it's really interesting to look at the proceedings of the 1973 meeting in Oxford, where everybody got together to talk about development and see how pessimistic, I've got the book in my bag if anyone wants to read it, how pessimistic the view was about the clinical advancement of cisplatin as a small molecule. And you can see that Hill summed up the meeting and said new and better analogs are awaited with great interest. And it was more of the tolerability of the molecule than its activity, of course, that was a concern at that time. And I don't know if anyone's read this book, but I also have it in my bag. But written by June Goodfield, who was a science reporter in the 1970s who attended the meeting and wrote a chapter about anti-cancer drugs called I've Got the Platinum Blues. And the reason that the chapter was called I've Got the Platinum Blues is because it was all about the cisplatin meeting. In fact, Barnett Rosenberg told her at the meeting that he believed platinum blues would ultimately end up being one of the clinical agents that went into humans and went into the clinic. And so there was a real uncertainty at that time between 68 and approval about what that agent would be, whether patients would be able to tolerate the drug in a form that it was administered to the patients. But obviously it got approved, and we're in an amazing place now, and I'm going to show you a couple of figures to give you a sense of just how impactful cisplatin has been to so many facets of the scientific research community and the clinical research community. So we've got a really amazing drug now. There are 3 approved anti-cancer drugs, multiple experimental therapeutics that have gone into the clinic. There have been clinical solutions that have been adapted to other clinical chemotherapeutic regimens since then as well. And of course, the combination chemotherapy implementation was a dramatic achievement as well. I mean, this synthetic inorganic chemistry, I say here, an entire field of that's created. The metals in medicine field really didn't exist. It was launched by cisplatin as well, yet another contribution to the community. And of course, that I think maybe the most impactful, but not so much reflected in this room, is cisplatin being used as a tool to understand the underlying cellular biology of cancer cells. And that's a really amazing utilization, and I'll give you a sense of how profound that is in a slide or two. And using cisplatin, it's driven everything from understanding lysosomal function, endo and exocytosis, DNA damage repair, the whole field of synthetic lethality that's associated with that. I know we'll hear a little bit about that later on. Uptake and efflux transporters, cell cycle effects, cell cycle regulators, metallothionein, glutathione synthesis, and drug deactivation pathways. None of that really existed when cisplatin was discovered. and cisplatin's been used to really demonstratively understand each of those cellular pathways. So really profound contributions across science because of the molecule beyond its amazing benefit to patients as well. So we've heard a lot about cisplatin and carboplatin today, and I really just put this slide here. I'm sure we'll hear about oxaliplatin later on, to remind people that outside the United States, there are actually 4 other approved platinum drugs as well. There are probably 7 that are approved for clinical use. Nitaplatin, which I show you up here, heptaplatin, lobaplatin, they're approved in China, Japan, and Korea, and miraplatin was approved recently in Japan as well. It's a really interesting molecule. There's a CH12, you can see here, there's a long hexane chain on both of these 2 leaving groups, and it's actually formulated in oil and injected into the hepatic artery directly to feed to the liver to treat hepatocellular carcinoma. That was approved, I think, 2 to 3 years ago in Japan. 2009, it says here, sorry, and then fully approved more recently. So the work still continues. I think you can all see the structure-activity relationships have not been violated, the fundamental structure-activity relationships in these sort of Me Too analogs of platinum complexes. I wanted to put these plots here to give you a sense of that impact of cisplatin. So I'm going to do that in a couple of ways. The first is publications. If you type cisplatin into PubMed, you get almost 70,000 publications, which is just stunning. And when you first look at this plot of publications per year that we've produced here, this is not cumulative. There are more papers about cisplatin every year than the year before, and that has always been that way. So this is absolutely remarkable, and the same is becoming true for carboplatin and oxaliplatin, which has got a lot more interest in the last few years as an experimental therapeutic. If you compare that to a lot of other DNA damage and agents that emerged into the clinic at the same time, they really flattened out. That passion really isn't there for those agents in the same way from a research point of view, and this is really an research impact perspective. And the only real comparators, we looked at a lot of drugs, Taxol and doxorubicin, which are also widely clinically implemented and still growing in terms of their research adoption and utilization. In fact, if you compare to something like imatinib, which was probably one of the reasons a lot of people had trouble getting cisplatin grants 10 and 15 years ago, even the publications on that really flatlined about 2006, 2007. So these targeted agents aren't really being studied nearly as intensively. The other way to think about impact is probably funding, especially for most of us. I think that's very true. This is a plot of grants per year from NIH, and there's some NSF data that's rolled into this, but it's extremely minor. The little tiny blue bars down there. It's just anything with cisplatin in the abstract. You can see got up to almost 200 grants a year at one point. This dip is really compelling. I know my PhD supervisor blames gene therapy on the lack of ability to get a grant about cisplatin around the turn of the millennium. And that seems to be reflected in the United States as well with this NIH funding. It's much harder to analyze dollars, of course, because of inflation and other reasons. The historical data is not complete, but I can tell you, for example, between 2009 and 2013, when it was pretty consistently funded at the same rate, that represents over $300 million in funding from NIH. So you can see there'd be easily over $1 billion in investment in cisplatin research that's gone out from NIH since its discovery, which is just absolutely stunning. And I'm not going to go into great details on this, but there are also a lot of quality-adjusted life-year analyses on the economic benefit of platinum-based drugs, which run well into the many, many billions as well. So any way you look at cisplatin and its discovery, it's just been an absolutely remarkable development for the wider scientific community, not just all of those hundreds of thousands of patients, if not millions, who've received platinum and had their life extended or received a total cure. So I wrote this paper for JMed Chem recently. It turns out it's really difficult to get the word damned into the title of a paper. That took quite a long time. It's a quote from Shakespeare, which is how I got by. It's referenced, so they couldn't shoot it down. But the damn nature reference was really to talk about the fact that there's a lot of need for research in platinum and for those improved platinum agents with new spectrum of activity, new scope of activity, and because it can be a difficult drug for patients to take, and a lot of patients still receive it. It's worth it, but it's difficult, and I'll show you at the end what I mean. So in that review, I talked a little bit about some of the opportunities for the field, and I think these are sort of areas that are fairly underexplored, and I'm just going to briefly mention each of those. Don't worry, I have my eye on the clock, so you stand up. So for the next 45 minutes, no. So the first one is imaging, and in fact, if anything's been very understudied, especially at a macro or a clinical imaging, a clinical level, it's been imaging. These are all of the images of patients with a radiolabeled platinum drug that has ever been reported and published, all of them. So there are 4 isotopes for platinum, multiple metal metastable isotopes that are amenable to SPECT imaging. You can see very early images back here of cervical cancer patient, astrocytoma, so this is a brain tumor. You can see pretty remarkable levels of drug imaged in that brain, and really from a personalized medicine point of view, imaging of drugs in the patient is something that needs a lot more attention for platinum drugs. There's some really nice high resolution recent, this is a healthy patient, this is a patient with metastatic lung cancer. You can see the drug in the tumors. It's really quite remarkable, and you can imagine how that could drive patient decisions. This is a PET imaging study using 13N labeled cisplatin. I spoke with the investigator that reported this study. Again, this is brain tumors. You can see over time the take-up of drug into these brain tumors. It has a 10-minute half-life, so it's literally a sprint from the PET radiochemical lab to the patient to inject it. But it's only been done once, and I think it's definitely worth a lot more exploration. And these are all cisplatin. There's no carboplatin, there's no oxaliplatin studies, and I think this is something we're thinking about. At a micro level, there's been a lot more work done on fluorescently conjugating to platinum. The weakness, of course, that I think is easily recognized is when you whack a huge fluorophore onto a tiny inorganic molecule, you modify it. So it's a qualifier that I think needs to be borne in mind. But when you do that, you can image them in terms of their cellular accumulation. You can do that quantitatively as well. This is some work last year with a colleague of mine at NIH where we were looking at ototoxicity, which is not really a dose-limiting toxicity, but a lot of patients walk away, especially from cisplatin chemotherapy, with tinnitus or deafness. And we're trying to understand why. It's long been known that patients 8, 10, 15 years after platinum chemotherapy are still urinating platinum at detectable levels. So you carry it with you for a long time. This study, we used this fluorophore-labeled version, and we could see cisplatin accumulating to quite high levels in inner ear cells. Not only that, when we took clinical samples from autopsy, you can see 18 months later, there's still quite high levels of detectable platinum in the inner ear. So it seems to hyperaccumulate in the inner ear and drive that ototoxicity. And so you can imagine a lot of work is needed to try and directly address the ototoxicity caused by cisplatin, perhaps with an adjuvant agent. I definitely think it's time for a new drug. I think there's lots of ways to do that. A lot of work in the 90s and 2000s has been driven towards trying to find a platinum drug that works against a platinum-resistant cell line. I would argue that based on everything we know about resistance, that's probably not going to be the ideal strategy. And I think the real benefit will come from looking for platinum drugs active against other tumor types that aren't currently treated. I think there's still lots of opportunity for Me Too drugs. Drug combination studies and screening opportunities for drug combination is a great way to win more opportunities for platinum in the clinic, and rule-breaking compounds. There are a couple of compounds that are in clinical trials in humans right now in the United States. PT112 is a phosphochelated drug, which is very unreactive from Bosa's group that's got a small company that spun out. There's a reformulation of carboplatin called dicycloplatin that just actually has an extra equivalent of leaving group in the formulation that's in clinical trials. And there's another drug which came out of Steve Lippard's work, which we don't know a lot about in terms of disclosures, but seems to have a maleimide dependent on a platinum 4 group that binds to protein albumin to really extend the pharmacokinetic half-life of the compound. The theory is that it would be reduced and releasing cisplatin with a much more prolonged AUC, so modified pharmacokinetics. And of course, there are lots of nanoparticle formulations that in one way or another attach a DAC or a diamine group through carboxylates to a liposomal formulation that ultimately releases the active agent. So there's a lot of work taking place there. But as I mentioned, there are drugs like brain tumors where I showed you those images. There's lots of platinum getting into those tumor cells. The ratio of carboplatin is about 10 to 1. I know a few years ago there was actually a satraplatin clinical trial for pediatric brain tumors that saw multiple responses, but then the study was closed down by the company because they had an alternative opportunity. So I think there's still, with the right people and a lot of energy, great opportunities for some of those experimental agents that were out there to find the right tumor for them and take them into the clinic. Of course, we all know now that in the recombinant protein stage, things like antibody-directed therapeutics are receiving a lot of attention. It's probably another wave of cisplatin grants that are really difficult to write. But there's been very little done on trying to conjugate a platinum-based toxin to antibodies. So traditionally in the antibody drug conjugate field, really toxic compounds abandoned in the 80s that were natural products that were extremely potent at cell killing, but also extremely toxic, are conjugated and directed straight to the tumor because of their antibodies' antigen detecting ability. So there are lots of these that have been in clinical trials. There are some that have been approved by the FDA already. There's only one paper that I can see where a platinum complex was conjugated to Herceptin, which binds to the HER2 receptor in breast cancer. It was shown to be taken up into cells. It was shown to be able to kill cells really nicely. And so I think there's a great opportunity in terms of bioconjugate chemistry expertise to explore antibody-drug conjugates with platinum as well and bring them back in another way. And I think it's really interesting when you get these fall-off rates in platinum funding, because some other new therapeutic technology comes along that everyone's excited about. What you usually find out later on is it's not nearly as good as everyone thought. Platinum stays in the clinic, and platinum probably works with that other thing to make it even better. So platinum stays the course no matter what. And then I'm not going to talk much about this slide, but with the emergence of chemical biology, there's been some, but not a huge amount of adoption of chemical biological techniques for platinum. And this is a really great example from the DeRose group, where they've been modifying actually picoplatin, another clinical failure that may have worked under in a different personality context. Picoplatin, where you can modify, for example, with an azido group that can you can create cross-link with something like a biotin for a pull-down study. And I think there's a lot of opportunities for studying the mechanism of action and the cellular targets of platinum. While I don't think anyone doubts that cisplatin kills cells by binding to DNA, someone pointed out to me last night that less than 1% of the platinum in the cell is on DNA. And so understanding all of the other things that platinum doing to the cells is a great opportunity. So for the last piece, I want to tell you a little bit about some work actually that Jim was quite involved in with me. It's a story that some of you are going to recognize because there's a lot of platinum chemists in this room. And so what I tell you, you'll know, but educating the entire community turned out to be important. So what this is a box of compounds. And I have a few boxes of these compounds, and they're all platinum complexes. And I don't know if I can say it, but I had the most amazing thing that happened to me is I wrote to Johnson Matthey about 6 years ago, and I said, my name's Matt, and I do drug screening, which is my day job that I'm not really talking about here. And I wish there was a collection of platinum compounds that I could screen against cancer cells. And one of their consultants wrote back and said, well, they're just in a cupboard here, all the compounds we ever made, and we've never really done anything with them. Do you want them? And I sort of like, yes, and I tried to be very measured in my response. And they didn't write back. And then 2 weeks later, I received 600 platinum complexes in powder form annotated. And they're in my office. And Jim has also worked with me to get some other compounds together for this. And we've been thinking, well, how will we do it? So if I walk into any lab and I said, test this compound for me and do a cell-killing assay, that student will put it into DMSO, and then they'll start their experiments. And a lot of people in this room know that DMSO is not a great idea. If there are any veterinarians in the room, cisplatin is actually used as a single agent to cure sarcoids in horses. And there's a lot of evidence, especially in Australia, of veterinarians or farmers just taking a DMSO solution of cisplatin, rubbing it on their hands and rubbing it on the horse because these are skin. So there's a lot of literature about that. These are the kind of compounds, I don't know which one this is, but I just took a photo there, number 358. So you can see all these boxes of compounds I've got. But what I knew, because I was trained in platinum, is that if you add DMSO to cisplatin, it doesn't kill cells anymore. This is clinical sample and saline sample. They perfectly overlay. Add DMSO, it doesn't work. But then I tried to reject a paper that I'd received where the people put cisplatin in DMSO, and the editor said, you can't reject it. Show me where it says that DMSO destroys cisplatin. So I sent them Sadler's and Lippard's papers showing the chemistry of the reactivity, but there's nothing about biological activity. So we wrote this paper, Say No to DMSO, and I'll show you why we did it. I took a couple of summer students and I said, go and read the most 35 recent papers on cisplatin in these great journals and write down what they dissolved their compound in. And here's DMSO and Cancer Research, wonderful journal. 34% of the papers, 12 of the papers, dissolved cisplatin in DMSO. And of course, probably just as concerning, 5 of the 35 papers were in clinical. That's wonderful. But 15 said not reported. And in fact, if you look across here, you can see that between DMSO and not reported, you've got from a third to a half of all of the papers using DMSO or not telling you what they're using for their cisplatin. So there's a real reproducibility problem here, especially if you extrapolate from 70,000 papers and wonder if a third of them are dissolving platinum in DMSO, especially all of those cancer labs that would treat it just like Taxol and every other organic drug. So we went methodically through, and in fact, there's in vivo use as well, people injecting animals with cisplatin and DMSO. So we went methodically through cisplatin, carboplatin, and oxaliplatin. This is exactly what people in this room would expect to see. If you dissolve your cisplatin in DMSO, it doesn't kill cells anymore. The same was true for carboplatin, and a very complicated story for oxaliplatin, but there was definitely effect on the biological activity. But it leaves us with this question, of course, if I've got 800 compounds and I want to dissolve them to do screening, what solvent can I use? And of course, we all know that even carboplatin and oxaliplatin are dissolved in water, and cisplatin is formulated in saline. So there's a lot of chemistry that's unique to each one of these complexes that will make it quite complicated. But this was an NMR that was collected with not cisplatin, but another platinum complex where we could do proton NMRs. And you can see that in the first 24 hours, all of the complexes completely reacted with DMSO and gone away. So we couldn't even make a rapid DMSO solution. the good news is, for us, is that we can use things like acoustic transfer now, which is really amazing. So you can take an aqueous solution of a compound, or 800 compounds, hit the bottom of that plate with your samples with sound waves, and a little droplet is fired up through the air. You turn your other plate upside down, and you just transfer your plates from a right way up to an upside down plate using sound transfer. And the droplets are of a perfectly known size. So you can see this acoustic transfer process. This would fly up and land on another plate. So it gives us an opportunity to think about how we're going to create this library and see if we can really go back and screen a lot of compounds that haven't received a lot of attention since the 80s against a lot of different cancer cell lines. This was supposed to be a video showing our robotic arms moving around, but the idea is to integrate them into our automated high-throughput screening platform. My day job's doing this kind of work normally. So we have a lot of fun. So in the final couple of minutes here, I want to comment on this. I think we're going to hear about it tonight, but when Trevor Hambly gave me these 3 slides that I've sort of adapted and used, you have this great clinical benefit. I'm going to make these jokes, but with reservation, but you've got this beautiful drug that's yellow, the yellow shirt of Neil Armstrong, absolutely amazing story. And I was always devastated when I saw this quote about receiving plutonium and how he cured it, because I remember thinking, oh, this was that chance for Cisplatin in Time magazine, and it didn't happen. But in fact, I did a lot of deeper reading to try and understand this, because it's kind of funny, but it also gives some context for patients and their understanding about the therapies that they're receiving and the implications. And in fact, in Armstrong's autobiography, he said this about cisplatin: a cocktail of 3 different drugs, so toxic the nurses wore radioactive protection. And of course, that was PPE that nurses are required to wear, because these are quite toxic compounds in a chronic exposure context, the way a nurse would see them during delivery to patients. So, but he actually thought that they were radioactive. So there's obviously some confusion about radiotherapy that he may or may not have received as well. But it gives you that sense of things. But in 2002, there weren't really many ways to find out about how a patient felt about their chemotherapy, or for a patient to even find out. But now, if my mum bangs her knee, she's on WebMD finding out exactly what it means and what she can do about it. And patients are a lot more informed now about the therapies that they're receiving. And I just wanted to point out that this JMed Chem would not let me publish tweets, it turns out. But I put these up here, because if you can just pick 1 or 2 and read them while I'm speaking. It's really easy now to go onto Twitter. If you type cisplatin, today it's just this meeting is all that everyone's talking about with cisplatin. But if you look historically, people talking about the drugs that they're receiving, how they feel about it, how it's impacting their family members. And you get these really personal narratives from people all the way through their chemotherapy and their treatment for cancer. And it's a way to get this really personal insight, it's public, but personal insight into the treatments that people are receiving, how they feel about it, the side effects that they may experience, but the joy of receiving these treatments, knowing what they will do for them and how they will help improve their life and cure them. So it's a truly inspiring drug. So there's an opportunity. I'd encourage all of you to go and take a look at that. So at good 28.5 minutes, I just want to say that one of the most amazing moments in my life was actually meeting Bernard Rosenberg in 2002 in New York at a platinum meeting. He told me, well, I'm sorry about the beer. I told him that I worked on Platinum 4, and he told me that Platinum 4 was actually his favorite. He was probably just being very kind to me, but it was really an amazing moment to meet him and with some of my other colleagues from the Hambly Lab and talk about Platinum for about 15 minutes, and it'll stay with me always. And just acknowledge my PhD supervisor at the University of Sydney and all of my colleagues at the National Cancer Institute and at NCATS, where I am now. Thank you very much for your time.
So we have time for 1 or 2 very short questions.
Matt, great talk.
Thanks, Mike.
With 3 drugs currently in clinical use worldwide, what is the attitude of NIH or NCI in terms of the possibility with these new Platinum 4 prodrugs of approving yet another platinum drug?
Yeah, it's a great question. I think activity will always win. And I think that the proof of that is that, when oxaliplatin was approved for use for colorectal carcinoma, it was a genuinely new use against a malignancy that wasn't receiving a platinum drug. And in combination with other agents, it showed superiority and it was approved. And I think that that's always going to be true. NCI still has a huge developmental therapeutics program where they look at experimental agents, just like happened in '68 with cisplatin, with much more advanced models. And so the National Cancer Institute, for example, now uses patient-derived xenografts, samples straight from patients into animals that they propagate from animal to animal. That was adopted a few years ago. But these are bioassays that are far more clinically relevant that didn't even exist 5 years ago in an NCI context. So I think that there's opportunities for old drugs to be re-examined, but also for new platinum drugs with new composition of matter to be assessed against those agents.
No other questions?
They're hungry.
I'm sure Matt will be happy to take other questions during lunch break and the rest of the day. So please join me in thanking Matt for his wonderful talk.
Thanks, Sang-Shu. So it is the lunch break. What I'd like to do, though, is let's thank all of our speakers this morning.